Dear centers, please, students, please report your attendance. We are beginning right away. Centers, please report your attendance. That's all I ask. All right. So, dear students, are you ready for the today's topics? I gave you some small sort of homework also. Okay. Hello, Shillong students. How are you? Other centers also, please report your attendance. Please do that quickly. Okay. So, yeah. Good morning, Senapati students. How are you? How are you doing? So, we had done a lot of part in the last class. We have basically completed the main parts of the light reaction. Okay. So, yeah. Hello, Itanagar students. Okay. Itanagar, you are the last one remaining. Please also report your attendance. I would ask you to quickly report your attendance. Yeah, please do that. Okay. So, centers, we are beginning the class now. So, let's start from where we left in the last class. You would remember that in the last class I had told you about photosynthesis. And regarding photosynthesis, let me give you a quick recap of what we are going to do today and what we discussed in the last class. I believe you also have received some notes that I gave you. Okay, that is a summary of the chapter photosynthesis. I hope you found that to your interests. Okay, yeah, I used a lot of references from the comics and the anime that I watch and the movies that I watch. Okay. All right. So you're asking me, yeah, Shillong, so you're asking me what's the difference between a lettuce and a cabbage. Both are of different genera. Both are of totally different genera. But let me ask you, are you asking me about sea lettuce or green lettuce, salad lettuce? Yeah. Which lettuce are you talking about? There is something called sea lettuce also. That is totally different. So, which lettuce are you referring to? Normal green lettuce or cabbage, I understand. That is Brassica oleracea variety capitata. But which lettuce are you referring to? Which lettuce are you referring to? Yeah, just quickly tell me. Both sea lettuce and the salad one. All right. All right. So, look, if you ask me about sea lettuce, that is an animal basically. That is a green alga, alva. Okay. The salad lettuce is totally different. The salad lettuce is a land plant. Okay. And when I talk about alva, that is aquatic. Okay. So one difference there goes right away. If two plants belong to the different genera, then they have totally different characters. If two plants belong to the same genera, they will have much more common characteristics. Okay. So that is the basic difference. All right. And especially the leaf one. Okay. I'll tell you, I'll just write this down because, look, I'll write that. You're asking me a doubt, so I'll just give a brief pause. Lettuce is Lactuca sativa. Lactuca sativa. A totally different plant. Okay. The leaves are edible, much softer. Okay. Cabbage. Cabbage represents a condensed system of nodes and internodes. We eat both in the nodes and internodes. And almost when you talk about Lactuca sativa, the leaves, the nodes, internodes, both are edible in both Lactuca sativa and Brassica oleracea variety capitata. But the thing is, cabbage represents a much more higher density, and the amount of phytochemicals that are present in cabbage are totally different. the cytochemicals present in the cell, they are totally different from lacteuca sativa. This is a basically thing is this family, the family of lettuce plant is Asteraceae, sunflower family. Sunflower family. And cabbage belongs to Brassicaceae or Cruciferae family. Brassicaceae family. Totally different families, not even of the same family. So you can understand that if two plants belong to a different family, then they have a totally different characteristic. Okay? Alright. So let us continue where we left off. I was teaching you about photosynthesis, right? So what is photosynthesis? Photosynthesis is making food in the presence of light. Photosynthesis is divided into two reactions, light reaction and dark reaction. But we will first begin with the history of photosynthesis. The history of photosynthesis starts with Priestley's experiment. So what did Priestley do? Priestley conducted an experiment, and in his experiment he basically showed that if I keep a mouse in a bell jar, inverted bell jar, the mouse will die quickly. If I keep a candle burning along with the mouse and candle both will extinguish, both will die. Okay, the candle will die as well, and it will extinguish, and the mouse will die as well. But if I keep a plant in sunlight, not in dark, then what will happen? Plant is giving oxygen basically. Now we know that. So basically the candle will burn for a longer period of time, and the mouse will remain alive for a longer period of time. Not for an indefinite period of time, but for a much longer period of time. And if I take so many plants together, then they will be alive indefinitely until the sunlight is coming in. Now tell me what will happen if I place the plant in sunlight and not in the sunlight but in the dark, and I keep a mouse and candle with it. If I take this experiment and I perform this experiment in dark, then what will happen? Okay, Shillong, thank you for the update. You have the attendance of all the centers now. Thank you, everyone. So, I have changed my question now. I am asking you, if instead of repeating this experiment in sunlight, what will happen if I perform this experiment in dark? Can you guess what will happen? Something very interesting will happen. Okay, no idea? Anyone? What do you think will happen? What will occur? All the three things will die. The plant will remain alive because of anaerobic respiration. Plants can perform that. But animals cannot perform anaerobic respiration, especially for a longer period of time. And the candle will extinguish also quickly. Okay. So basically everything inside that bell jar will die. Because the plant does not receive oxygen, the leaves will, cells will keep on dying quickly, one by one. Some cells might remain, so the plant might regenerate after some time, but most of the cells of the plant will also keep on dying. Okay, so if you perform this experiment in the dark, now in dark, the plant is not giving oxygen, it will give only carbon dioxide, because it is also consuming oxygen. All three things are oxygen consumers at dark. Only in the sunlight, plant gives oxygen. Alright, continuing. So Priestley's experiment proved that plants give pure air and take in impure air. Animals while breathing and substances while burning release impure air. Impure air is carbon dioxide. But nobody knew exact composition at that time. And pure air was later described by Priestley as oxygen. Okay, oxygen means life-giving. Gen means to give, oxy means to give life. So oxygen was simply something described by Priestley that gives life to organisms. Then there was a scientist called Nicholas de Saussure. Saussure proved that amount of impure air taken in by the plant, it is basically equal to the amount of oxygen released by the plant. So if plant is taking in carbon dioxide and giving out oxygen, so people will assume that the raw material of photosynthesis is carbon dioxide and basically carbon dioxide and water, है ना? So carbon dioxide and water will be the raw materials, and if you make the products, what is the product that you are getting? You are getting food. Food is made in the form of glucose or starch. Starch is basically polymer of glucose, so everybody knew this. So because how did everybody know that starch is a polymer of glucose? It's a very, very simple experiment. How will you know that starch is a polymer of glucose or it is a polymer of sugar? Everybody can perform this experiment, and it will not take more than 30 seconds to prove that starch is composed of sugars. And the experimental material is the thing you will perform that experiment is on. It is right there in your class. How will you prove that starch is composed of carbohydrates? And I'm telling you that you have all the things needed to perform that experiment very close to you in your class. Can you guess? Take a wild guess. How will you prove that starch is composed of sugar? Later on, people wrote this equation that carbon dioxide, water gives starch or glucose plus oxygen. I'm telling you, you can perform this experiment right now. Yeah, very good, exactly. Digestion. Not even the first step of digestion. Take a piece of bread, put it in your mouth. I'm very happy. Who has answered this question? Yeah, please write your name. That's very sharp of you. Very good observation. Yeah, I'm happy that you answered. Shem, okay, very good, Shem. I appreciate your sharpness for this. Yeah, take a piece of bread, put it in your mouth. After 30 seconds, you will keep on, if you keep on chewing that starch or any chapati or anything which contains starch material, you will keep on experiencing the sweetness. Even bread, after chewing after some time, you will experience the sweetness. Why? Because there is an enzyme called maltase in your saliva, and maltase digests starch into a dimer, a disaccharide of glucose that is called maltose. If I digest maltose further, it will give glucose. Okay, so maltase is basically used in beer making, wine making, so that everybody knows that basically starch is composed of sugar, right? So very good, Shem. I'm very happy that you answered quickly and very fast also. That matters. So keep it up. I'm happy. All right. So basically, carbon dioxide and water, they are giving glucose and oxygen. So this was the first equation of photosynthesis that people understood. And because Saussure is saying that amount of oxygen is equal to amount of carbon dioxide, so people assume that carbon dioxide molecule is being broken down and oxygen is released from it. Everybody will assume it, right? But then it was proved that it is not true. There was a scientist called Van Niel, and Van Niel basically proved that if you take this equation, then there is a change in the equation that is needed to be done. And how did he find out this change? He observed sulfur bacteria. And in these sulfur bacteria, what happens is they take carbon dioxide and they take hydrogen sulfide also. And hydrogen sulfide is taken in. This hydrogen sulfide, if suppose you are making oxygen by breaking down carbon dioxide, then in this reaction also you should get oxygen, but there is no oxygen given out here. So what is being given out? It is sulfur. So this means that hydrogen containing substance, which is called hydrogen donor, that is being broken down. Alright. So hydrogen donor breaking down was proved by Van Niel. Okay. But this was basically just an estimate. So nobody gave this a confirmed nod that yeah, this is confirmed that hydrogen donor is being broken down. But it was a guesswork Scientists guess a lot of times, okay? Basically, science is all about theory, and very few things in science are being proven. So, proof requires very much time. So, not everybody believed Van Niel, but because of this development, the equation of photosynthesis becomes 6 CO2 plus 12 H2O gives glucose plus water plus oxygen. Now, please observe, we are putting 12 H2 on this side and 6 H2 on this side because of Van Niel. It is still not proved confirmatory. There is no sure shot proof up till now that definitely oxygen comes from water and not from carbon dioxide. Oxygen comes from hydrogen donor, not carbon dioxide. So we needed a proof, all right? So and this proof was provided by which two scientists? Rubin and Kamen. So please write now. Today we will start with Rubin and Kamen's experiment. Rubin and Kamen experiment. These two scientists proved that Rubin and Kamen proved that oxygen in photosynthesis comes from photolysis of water. Okay. So what was their experimental material? Does anybody know? What was their experimental material? All right, I'll write it down for you. They used chlorella, a green alga. They used chlorella, a green alga, using H2O18 as hydrogen donor. Can anybody tell me what is this? Is this heavy water or is it something else? Is it heavy water or is it something else? H2O18 or oxygen 18, basically it's a heavy isotope of oxygen. So tell me, is this heavy water, yes or no? Yeah, it's a heavy isotope, but can you call it heavy water? Can you call it heavy water, yes or no? I'm just testing you. It's not heavy water. It's not heavy water. Heavy water is totally something different. Heavy water is D2O, deuterium oxide, okay, or you can call it di-deuterium oxide. Normal water is called diprotium oxide, okay. So it is not heavy water. Heavy water we will call it only when hydrogen is not protium but it is deuterium, all right. So this is not heavy water, but it is water containing heavy isotope of oxygen. And it is non-radioactive. Please try to remember. Water containing heavy isotope of oxygen. Water containing heavy isotope of oxygen, and this is oxygen 18. Okay. Now I will modify the reaction of photosynthesis, and please observe what is going to happen. So aquatic alga is kept in H2O18 and it is supplied with carbon dioxide, normal carbon dioxide, 6 CO2 plus 12 H2O18. Now because I am using a heavy isotope, so I will just use a different color to represent it. I hope that you understand this. H2O18. Alright? 12 moles of or 12 molecules of H2O18. For oxygen, I am using a different color. Let me take some color like, I will take this color. Okay. So what is going to happen? I am going to get C6H12O6 normal glucose plus I am going to get 6 oxygen 18 is evolved. It is being released and non-heavy normal type of water. Ultimately, all the H2O18, it will be released as oxygen 18. None will remain in the solution. So this proves that H2O18 is the is the hydrogen donor. And how will it give hydrogen? It will be split into two halves. Hydrogen will be separated. Oxygen will go on the other side. Oxygen will be evolved as gaseous oxygen. Hydrogen will be absorbed by the carbon and it will be reduced. So basically, carbon molecule is being changed into something else. Oxygen is being changed into something else. Okay. Now please tell me, are you familiar with the concept of oxidation reduction and oxidation state? Just raise your hands if you are familiar with all these three things. Do you understand oxidation reduction as well as oxidation state? Yes, everybody remembers? Okay. So please look at this equation and tell me which quantity, which substance or which element is being oxidized and which is being reduced. Or is it only oxidation, only reduction, both? Photosynthesis is, question, photosynthesis is a reduction process, oxidation process, both 1 and 2, fourth, none of the above. Now, I just don't want the answer only. I don't want the answer only. I want you to tell me which element, which species, which atom, which radical, which molecule, whatever that is, it is what you want to say, it is being oxidized or it is being reduced or it is being both. Can you calculate? Can you calculate the oxidation state? If you can calculate the oxidation state, you will know the answer. So this is the equation right in front of you. And I want you to tell me that which species is being oxidized, which is being reduced. I hope everybody has written. I'm sorry if I scrolled it too fast. Okay. On the basis of this reaction, it was proved that one species, this was the conclusive proof. And after this proof, I want you to tell me where is oxidation happening, where is reduction happening, or where are both happening. And I also want you to give me the answer. Sorry, this is the third option. My bad. This is the third option. Can you do that quickly? I'm waiting for your answers. I'm waiting for your answers, students. All right. No answers yet. You want me to solve it? If you are having difficulty, say yes, we are having difficulty. Please solve it quickly. I'll solve it for you. I will solve it for you. Okay, you are taking some time, so I will solve it for you. Look, this equation was used by Rumen and Kamin, that oxygen 18 is converted into oxygen gas. So let me solve this question for you now. I'm writing the normal equation of photosynthesis now. 6 CO2 plus 12 H2O. I'm not writing 18 now because it is not required anymore. I'm writing the normal equation because not every time I will use oxygen 18. It's a very rare isotope. It normally does not occur in nature. So now it is going to become C6H12O6 plus 6 oxygen plus 6 H2O. Now please take a look at the oxidation state of carbon and oxygen. What is the oxidation state of carbon in oxidation state of carbon in CO2? What is the oxidation state? I will write carbon dioxide. Look, carbon dioxide is a neutral molecule. So neutral molecules have no charge. This is a gas. Suppose, because I do not know the oxidation state, I will write X. X is the oxidation state of carbon in CO2. Minus 2 is the most common oxidation state of oxygen. And there are 2 molecules of oxygen. So I will write 2 multiplied by minus 2 equals to 0. So this is a bit of chemistry involved now. X plus minus 4 is equal to 0. So basically this will become X minus 4 is equal to 0. X minus 4 is equal to 0. This means X is equal to plus 4. So the oxidation state of carbon in carbon dioxide, it is plus 4. Now please tell me oxidation state of carbon, oxidation state, in short I am just writing OS because it is becoming very long. So I am just writing OS. OS, oxidation state of carbon in C6H12O6. Can you calculate? Suppose it is X again, or you want to say Y. So suppose it is Y. Okay. So suppose this is Y. Hydrogen's most common oxidation state is plus 1. Oxygen's most common oxidation state is minus 2. So my equation becomes, because glucose is a neutral molecule, so the total charge on glucose is 0. So now I am calculating oxidation state of carbon in glucose. So it becomes 6Y plus 12 into plus 1 plus 6 molecules into minus 2 equal to 0. Right? Now I will solve this equation. 6Y plus 12 minus 12 is equal to 0. 6Y is equal to 0. So what is the value of Y? What is the value of Y? 0. Exactly. Good. Don't say infinity. I hope nobody says infinity. Because if I write 6 upon 0, then it will become infinity. Any number divided by 0 becomes infinity. But if you divide 0 by any number, the answer is 0. So this is basic algebra. Okay. So I will write the oxidation state of carbon is 0. Alright. So if the oxidation state is changing from positive 4 to 0, that means, is it oxidation or is it reduction? If oxidation state is changing from plus 4 to 0, is it oxidation or is it reduction? Anybody? Is it going to be called oxidation or will we call it reduction? Okay, I'll give you a formula for remembering these things. Oil rig. Oil rig. Oil rig means oxidation is loss of electron. Very good. Yeah, you answered it correctly. Reduction is gain. Whenever you forget it, try to remember this. Oil rig. Oxidation is loss of electron, reduction is gain of electron. Alright. So plus 4 will become 0 when it gains 4 electrons. So this is going to be reduction. So I am going to write carbon is getting reduced. This is reduction. Okay. Now consider oxygen. Okay, why should we consider oxygen? Because on the right hand side, oxygen is in 0 oxidation state. Watch here. This. Suppose the oxidation state of oxygen is Z. Okay. Now I will write total charge on any gaseous molecule, any element, any molecule which is neutral is 0. So I will write 2Z is equal to 0. This means Z is equal to 0. 0 oxidation state. Now see on the left hand side, in carbon dioxide oxidation state of oxygen is -2. But what about water? What about water? Water is here, right? Here it is -2. In fact, whenever we talk about oxygen, on the left hand side it is always -2. Yeah. Only in peroxide, superoxide will oxidation state not be -2. Okay. Now, some people can say, sir, why is it not disproportionation reaction? Because in carbon dioxide also oxygen is there. So some people can ask me this also. The thing is, if carbon dioxide was disproportionated, then we should not include water. Okay, but we require water for balancing the equation. This means that oxygen is definitely coming from water molecule only. Or also Rubin and Kamen has proved that oxygen gaseous is coming from water. So you will not take oxygen from carbon dioxide. You will take oxygen of the water. Here it is -2 and here it is 0. So please tell me, is it reduction or is it oxidation? The oxidation state of water, it is -2 oxygen, and in oxygen gas it is 0. So it is oxidation or is it reduction? I have just written for you oil rig. Oxidation is loss of electron, reduction is gain of electron. Oxidation state is changing from -2 to 0. Is it loss of electron or is it gain of electron? Please answer quickly. Shem, will you answer? Other students also, please answer quickly. I believe maybe there is a lag in the chat, so that's why your answers are getting late in reaching to me. Alright, so I guess there is a lag in the chat. Okay, I'll tell you. I think that everybody must have guessed it by now. It is oxidation. Why? Because -2 will become 0 only when it loses 2 electrons. So there is gain of electrons, reduction, and there is loss of electrons, oxidation. Both processes are happening. So carbon is getting reduced, oxygen is getting oxidized. So the photosynthesis becomes both a reduction process and a oxidation process. So my answer to this question is both 1 and 2, both reduction and oxidation process. And now you can write photosynthesis is a photochemical redox reaction. Photochemical redox reaction. Clear to everybody? Clear to everybody, right? So this is the photochemical redox reaction. Photochemical redox reaction. So this is clear to everybody. Any doubt about it? Shillong, Itanagar, Senapati, Shillong. Is there any doubt regarding this now? Is there anything that you did not understand about this? Okay, so let us, no doubt. Okay, very good. We are happy. So let's move further. All right, very good. So let's move further now. Photochemical redox reaction is over. So this is what I wanted to tell you, everyone. So now we will move on to the further part. How is carbon getting reduced and oxygen is getting oxidized? Now, as we move further, we need to clarify a few little things, and some of these things include there is a very famous experiment regarding what this process is doing, and it is called action spectrum of chlorophyll determined by Engelmann's experiment. Engelmann's experiment. Okay. So we need to discuss this also. Action spectrum of chlorophyll. Please put the heading. Action spectrum of chlorophyll. What is chlorophyll? Why do we want to talk about it? There is an experiment that proves that chlorophyll is necessary for photosynthesis. How many people of you do not know about this reaction, that chlorophyll is necessary for photosynthesis? Everybody knows, right? Okay, if you don't, let me remind you. There is an experiment in which what we do is, we take plants which have variegated leaves. Okay? Just a second. Okay. Variegated leaves, you know, plants which have yellow patches or red patches like this, or white colored patches. Okay, there are many plants like that. Okay. These are called plants with variegated leaves, right? If we take such a plant, take a leaf of this plant and because we want to prove that chlorophyll is necessary for photosynthesis, so I'm going to prove that plants which have such leaves, only the chlorophyll containing part will synthesize starch. So first I will take this plant, keep it in the dark for 2 days, then I'll take a leaf of this plant and I will put it in sunlight for 2 hours. After putting in dark for 2 days, the purpose of keeping in dark is because every cell of the leaf contains starch molecules, because starch is being transported from one part to the other. So first for 2 days I need to keep it in dark so that starch molecules become exhausted. After that I will keep it in sunlight for 2 hours, and then I will take the leaf and I will perform starch leaf experiment. Only the green parts of the leaf will show blue color, the red and the yellow or white colored parts, they will show no color, no blue color. So that means they will not be synthesizing starch because they do not have chlorophyll. So basically, scientists, everybody at that time understood that chlorophyll is necessary for photosynthesis. Now I need to study about chlorophyll. I need to study about the spectrum of chlorophyll, right? So let's write quickly. Action spectrum of chlorophyll. What is chlorophyll doing? In detail I will discuss later, but let me tell you that chlorophyll molecule molecules are light capturing pigments, and chlorophyll is not the only pigment found in leaf. Chlorophyll is not the only pigment found in leaf. So what are the other pigments found in the leaf? What are the other pigments found in the leaf? Can you guess? Carotenoid, xanthophyll, all these pigments are found in the leaf, right? So carotenoid, xanthophylls, they have a different color than chlorophyll, that is non-green color. Okay, so let me tell you about it. Chlorophyll occurs along with carotenoid and xanthophyll pigments and all these pigments are arranged in the form of a group. Okay, all these molecules are arranged in the form of a group. So let me tell you about this group. This group is called light harvesting complex or light harvesting antenna. Just give me a second. Alright. So basically what is happening, this is a light harvesting complex. It is called quantasome. Okay, and it contains of light capturing pigments which are transferring their energy finally to the chlorophyll molecule. This is what I'm drawing here. And why I'm discussing action spectrum of chlorophyll along with this, the reason is chlorophyll molecule absorbs light in a different direction, or basically it absorbs light of a different peak wavelength other than carotenoid and xanthophyll molecules. Okay? And how is this happening? Let me show this to you. I have given you a small summary. I think you will find some interesting things in there. And on the top of this is a single chlorophyll molecule. Here, chlorophyll A. All the other pigments, chlorophyll B, carotenoids, xanthophyll, they will be present behind this chlorophyll A molecule. So this is called light harvesting complex. Light harvesting complex. And light of different wavelength, blue, green, red, okay, every wavelength is falling on this light harvesting complex. Okay. Right. So all the other color wavelengths, they are basically falling on this. And why chlorophyll molecule is present in the end? Because it is by a phenomenon called inductive resonance. You can understand this, that just as a funnel collects substances, this is a light funnel. It focuses all of the energy onto a single chlorophyll molecule. Okay. So all the different wavelengths of light that are entering here, they are basically collected, harvested, okay, and they are transferred to the chlorophyll molecule. This phenomenon is called as inductive resonance. Inductive resonance. Inductive resonance. This is a chlorophyll A molecule. So these are the antenna molecules. Antenna molecules. Everybody knows what an antenna does. Antenna collects satellite signals. It collects different signals, amplifies them. Lot of signals are being collected, then only you can amplify it. Otherwise, signal strength will become very weak. So this is an antenna complex. Okay. And this is how the pigments are arranged together. Okay. These are carotenoid and xanthophyll pigments. Carotenoid carotenes are found in carrot, if you remember. Carotenes, if you go a single oxidation step, it converts into vitamin A also. Carotenes and xanthophylls. Xanthophylls called accessory pigments. These are known by another name, accessory pigments. Okay, these are called accessory pigments. Accessory pigments. This total thing this total system, it is called a quantasome. It is also called antenna molecules, antenna complex or antenna molecules plus chlorophyll A. It is called photosystem or quantasome. Photosystem is a more accurate term because we will be using this term repeatedly when we study the light reaction. Okay, so that will come handy there when I will remind you that there are two types of photosystem depending upon the type of chlorophyll. For now, try to understand that this whole complex, it is called photosystem. Okay, and it will capture sunlight, and chlorophyll molecule will get excited because of it. Okay, so I'll discuss that also. Okay. So this is done, right? So let me continue further. Now, there was a scientist called Engelmann, and he devised an experiment to determine the action spectrum of chlorophyll, which means that which wavelengths are absorbed more strongly and which wavelengths are not being used by the chlorophyll molecule. All right. So let us discuss that. T.W. Engelmann demonstrated the action spectrum of chlorophyll, using a simple arrangement, using a simple arrangement. Okay, do you need a break or something? Let me know quickly. I'll give you a 5-minute break because, yeah, for that, now there is another 1 hour. So I think you can take a 5-minute break now, if you want it. Yeah, because it's 1 hour now. Yeah, take a 5-minute break, okay? And I'll continue after that. Yeah, please just be very quick. I forgot to give you the break. I just remembered, yeah, that it's 1 hour now. Okay, so take a quick 5-minute break, and we'll be back after that. Okay. Okay, so break is over. Okay, so let's get back quickly from where we left. All right. So I was telling you about Engelmann's experiment. Engelmann was a scientist, and the technique that he used to demonstrate his experiment is considered a truly genius, remarkable experiment of his time, because Engelmann had very few resources at that time, and using those resources, he very cleverly managed to show what was not able to be demonstrated by any technology that was available at that time. There is no technology that could have exactly helped Engelmann at that time, because at that time, not even lasers, not even spectrophotometer, the things that measure the absorption of light, those were not invented as of his time. But still, he was able to show it with the help of a very indirect approach, which proved what kind of a genius he was. So let me tell you what Engelmann did. He chose an alga, okay, a non-motile green alga called cladophora. So you can see cladophora here. It's a non-motile green alga here, right? And he was using this green alga to prove something. So let me demonstrate to you what he did. He took a glass slide, okay, and he coated it with green alga, the cladophora. Now, cladophora is non-motile, so that means it cannot move from one place on the glass slide to the another. So everybody can see this. These green dots mean I am referring to cladophora, the green alga. Now what he did, after coating it with green alga and drying it, a very slight drying was done. He put it in the top of a flame and removed it quickly. So alga did not die, but they stuck to the glass surface. Okay, so they stuck to the glass surface. And now what he did, he put some motile aerobic bacteria on this glass slide. This glass slide contains two things. Motile aerobic bacteria were spread uniformly. Motile aerobic bacteria, they were spread evenly, they were spread uniformly. And the other thing, what is it? These are non-motile green alga cells. Of cladophora. Of cladophora. Right? Now he put this experiment in sunlight. If you put this in sunlight, all over the glass slide there is uniform deposition of green alga. So bacteria are dependent upon oxygen, so they will be spread all over the glass slide evenly. If you put sunlight, if this glass slide in sunlight, okay, so what is going to happen? You will say that in sunlight, when you place it in sunlight, then there is uniformity. Okay, the alga are spread uniformly because alga is going to perform photosynthesis, and photosynthesis releases oxygen. So oxygen is used by bacteria, and if uniformly oxygen is coming, then the bacteria will be spread uniformly. Okay? Uniform distribution of bacteria in sunlight. Okay? But when you put it under a glass prism, then a spectrum, Vibgyor, violet, indigo, blue, yellow, orange, red, it will fall on the glass slide. And Engelmann said that if I want to determine that which color of light is most effective for photosynthesis, then he said that I will be able to determine that in which direction bacteria are going. Bacteria will go in that direction where more photosynthesis, more oxygen is evolving. This will give an exact idea which color light is best for photosynthesis. Right? So let us perform this experiment. Second part, the main part of this experiment. I'm going to use a glass prism over this experiment. So give me a second. I'm going to take this experiment and I'm going to perform it by putting a glass prism in between. Okay, there was a, okay, some parts have not appeared over here. All right. So I'm going to put a glass prism here. Sunlight, glass prism. So when sunlight passes, white light passes through a prism, it gets split into 7 colors. I'm going to use these 7 colors now. Give me a second. I'm placing the sun on top now. I hope nobody gets, yeah. All right. So this is the sun. I'm putting a glass prism in between. So when white light passes through the glass prism, it gets split into the 7 colors. I'm going to just want to show refraction to you guys. It's not getting worked properly. Right. So when light enters the glass prism, it is going to split into 7 colors. I'm calling it violet, indigo, blue, green, yellow, orange, red. Okay? So what is happening? This spectrum of colors, it is basically falling on this glass slide. Right? So I guess everybody finds this much prettier than you were expecting. Yeah, all right. So let me continue. orange, yeah, and red. So I put the spectrum here. So when you put this spectrum over here, I've used all the 7 colors accurately, right? So it must be easier for you to identify which colors are which. Violet, indigo, blue, green, yellow, orange, red. Every color is accurate, entirely accurate, I must say. So let me widen this orange color a bit, because then only this will be complete, right? Orange has become a bit suffocated. Yellow is very less. Actually, yellow is also a very good part of the spectrum. So let me widen it. Then orange color, and then finally red color, right? So the spectrum is complete now. And now please tell me, what do you expect that is going to happen on this glass slide? I have split the white light into 7 colors, and this light is falling on the glass slide. So what do you expect will happen on the glass slide? Different rate of photosynthesis, right? These are 7 colors. Each color will cause photosynthesis, or it will not cause photosynthesis. And I can say basically that what is going to happen if these colors are producing the photosynthesis, then because of the photosynthesis, each will have a differential rate of photosynthesis. And I want to measure this differential rate of photosynthesis. Okay, I want to measure this differential rate of photosynthesis. Just a second. So let me tell you what is going to happen here. When this glass slide was previously placed, when this glass slide was previously placed, what was happening is the bacteria was spread uniformly over the glass slide. Okay, the bacteria were aerobic, and because of sunlight is spreading evenly. Sunlight has all the colors. Even rate of photosynthesis was there. So all the bacteria, they are motile also, but they are getting equal amount of oxygen. So there is uniform deposition, uniform rate of distribution of bacteria. Now it is not going to be the same. There is going to be a change, because the 7 colors will produce a different effect, and this different effect is what I am going to tell you now. Based on this experiment, Engelmann saw that there Engelmann saw that there is a different rate of photosynthesis. I'm going to erase some things now, so please pay attention. Okay. All right. So let us see what is going to happen. The bacteria will go towards that side where more oxygen is going to be present. Now you have already learned that algae are spread evenly like this. Okay, so algae have a uniform deposition. Now because algae have a uniform deposition, it is very difficult to show it by a different color. So now I'm going to just show you the distribution of bacteria. Distribution of bacteria is shown by red color, but red is already in the spectrum, so let me show different color. Suppose this color is for bacteria. Blue color region and red color region, maximum bacteria will be found. Maximum bacteria, they will be found in blue colored region, red colored region. And in red colored region, the density of bacteria is very, very high. In blue region, in blue region, the bacteria is still present, but in red, more bacteria are present. And in green region, there is no bacteria at all. In green region, there is no presence of bacteria at all. Only alga is here. There are no bacteria. Bacteria have gone over either towards the blue side or the red side. Okay. Bacteria are going either towards the blue side or the red side. Clear? So this is what I wanted to show you. Engelmann saw that. Engelmann placed a glass slide containing aerobic motile bacteria and immotile green alga, immotile green algae under Vibgyor spectrum of prism. Under Vibgyor spectrum of prism. So what happened was, under Vibgyor spectrum of prism, and he found that maximum bacteria were found in red region, were found in red color region, followed by blue color region, followed by blue color region. Okay, and in green color, no bacteria were found. In green color region, in a region of algae, illuminated by in region of algae illuminated by green light, no bacteria were found. In region of algae illuminated by green light, no bacteria were found. Okay. No bacteria were found. So this means, why did he do this experiment and what is the result of it? Engelmann performed this experiment because he wanted to show that there is different rate of photosynthesis in different color of light, and because in green color there is no bacteria, there is no oxygen. This means that there is no photosynthesis in green color light. Are you getting me? Why are plant? There is a question that is asked by class 9th, 10th standards. Why are plants green in color? Why are plants green in color? So the answer is, why the plants are green in color? Because chlorophyll does not absorb the green color light. Chlorophyll does not absorb green color light. It absorbs all other color lights, but it does not absorb green color light. Are you getting me? So that is why there is no photosynthesis in green region, because it is not absorbed at all. It is not used at all. So it is going to be reflected. This is why plants are green in color. Amount of oxygen evolved, amount of bacteria is proportional to amount of oxygen and amount of oxygen is proportional to amount of photosynthesis. So this means, because all these three quantities are linked together, this means amount of photosynthesis is directly proportional to amount of bacteria. So wherever there are more bacteria, more photosynthesis; no bacteria, no photosynthesis. You can say more bacteria is equivalent to more photosynthesis, and no bacteria means no photosynthesis. Alright. So he concluded that because there are no bacteria in green light, so there is no photosynthesis occurring in green region. So on the basis of above experiment, on the basis of Engelmann's experiment, following results can be determined. What are these results? What are these results? Can you tell me by yourself? What are these results? Anybody? Would you like to answer by yourself? There are two important results. Maximum amount of photosynthesis occurs in red light, followed by blue light, and no photosynthesis occurs in the green region. These are the results that I am writing down for you. Maximum photosynthesis takes place in red region. In red region of visible spectrum. Of visible spectrum, followed by blue region, okay, and there is no photosynthesis in green region. There is no photosynthesis at all in green region of sunlight. This also means chlorophyll is green in color because it reflects green and absorbs all the other colors. Because it reflects green light. Is this clear to everybody now? This is Engelmann's experiment. This is Engelmann's experiment. So is there any doubt regarding Engelmann's experiment? Everybody has understood about Engelmann's experiment? It is clear to everybody, right? Say yes or no. Okay, I think maybe the chat is getting delayed or something. All right. I guess if there is any doubt, let me know. Okay. All right. There must be a internet issue, I guess. Okay. So this is regarding the Engelmann's experiment. Now you have studied about the Engelmann's experiment. You know which color of light is best for photosynthesis, red and blue. So basically, except green, all colors are good for photosynthesis. This is a very important part. Now that we know that chlorophyll is using these colors of sunlight for making food, making something out of it, we need to understand what photosynthesis is, and we need to understand the various steps involved in photosynthesis. So now our part becomes to divide the light reaction and the dark reaction into their further steps, and we are going to study about it now. All right? Okay, you did not copy. Okay, yeah, my bad. Sorry. I'll just scroll up. Let me scroll up. Okay. All right. So please copy it, and then we'll move on further. Yeah, okay. Copy it. Let me know when you're finished. Copied? Okay, great. So this is about the visible region of spectrum. You know that there are two regions of light. Yeah, sound is not clear. I'm not sure what is wrong. Is it clear now? Taming students are saying. Is my sound clear to everybody? Centers Itanagar, Shillong, Senapati and Taming. Is it clear now? I'm not sure what's the problem. Okay. Yeah, just reply to me when you can. Okay, thank you, Itanagar. So my sound is clear to you. So Taming, just ask the technical team to see what's wrong. There could be an issue on your end. Okay. So now let's begin photosynthesis and the steps of photosynthesis, what is happening and where it is happening. I need to discuss this with you. Photosynthesis consists of two phases. The first phase is the light dependent phase. Light dependent phase. Right? So it is depending upon the sunlight. Right? So light dependent phase is also called the light reaction. Light reaction. Light reaction is sometimes also called Hill's reaction. I will discuss what Hill's reaction is when I finish light reaction. Then there is the second phase of photosynthesis, and the second phase is called the light independent phase. Light independent phase or dark reaction. Or dark reaction. Okay. So light reaction and dark reaction, these are basically the two phases. And first of all, we will take the light reaction, and after that we will discuss the dark reaction. So let's put the heading light reaction. The first phase, you can say photosynthesis, is divided into or consists of two phases, the light independent phase and the light dependent phase. First, it is the light dependent phase, then comes the light independent phase. Now I will tell you where it is happening. I have told you that there are two phases. Now I must tell you where, what is the location. Light dependent phase happens in the grana of chloroplast. Where? In the grana of chloroplast. Grana, everybody remembers? Those stack of coin-like structures that are there. Grana and grana thylakoids. They are the place of light reaction. Okay? Where is light independent phase happening? It is happening in the stroma. Stroma of chloroplast. In stroma of chloroplast, and it is happening in grana of chloroplast. All right. in the grana, in the stroma. So these are the two sites of light and dark reaction. So let us study what is the light reaction, and then we'll move on to the dark reaction. So what is light reaction? Considering the time that I have, I won't be able to say too much, but let me give you a quick introduction what is happening in the light reaction. I have hardly about 10 minutes or less. So light reaction, you must ask me this. So how were the two phases that there is a light reaction, that there is a dark reaction? How were they discovered? So basically, there were some scientists and they performed photosynthesis, or they allowed photosynthesis to happen in intermittent light. Okay? Intermittent light means you take light, you're giving light and then you're switching it off. Alright? Giving light in intermittent phase results in higher rate of photosynthesis. Okay? And these were the reasons that people suspected that there must be two phases. One must be a light dependent phase and other must be a light independent phase, so that when we are cutting off the light, that means the plant is quickly using up whatever it has prepared and put it into the dark reaction. And then when switch on the light again, light reaction begins again, dark reaction begins after the light is cut off. So in intermittent light, it is reported that there is a higher photosynthetic yield. So based on that, people guessed that there is such type of a reaction. Dark reaction was basically discovered by Calvin and his associates using chlorella green alga and a lollipop apparatus. Lollipop apparatus I will discuss later. It will take quite a lot of time to understand what lollipop apparatus is. But light reaction is comparatively quite easy. Light reaction, there is two parts of light reaction. In the first part, you want to make two things, two very important things. One is source of energy and other is the reducing agent. Look, photosynthesis is an energy using process, endothermic process. And dark reaction or light reaction, whatever you want to call it, photosynthesis is basically energy utilizing process. Respiration is an opposite process of photosynthesis. It is energy releasing process. So if you are making something which is energy rich, you require energy. That is clear to everybody? So first of the thing that we require to make glucose, it is energy source. The second thing that we require, that is a reducing agent. Reducing agent is NADPH2. I'll discuss that with you later. But why do you require a reducing agent? Because a reducing agent that is required to reduce carbon dioxide into glucose. What is carbon dioxide? C double bond O, C double bond O, CO2, right? And what is glucose? CHO, CHOH, CHOH, and then sixth carbon has CH2OH. 6 carbons, CHO, CHOH, CHOH, and CH2OH. So that means what is happening here? Carbon dioxide has 2 oxygen, fully oxidized carbon, and this is something reduced because it contains hydrogen. So you are requiring reducing agent. I have also shown you that carbon oxidation state. On the basis of that also you can explain that carbon is undergoing reduction. So I need a reducing agent to reduce the, CO2 into glucose. That is why I require NADPH2 and I also require energy. All these things will be manufactured in the light reaction. Okay? So light reaction is used to manufacture two important things, and one product is a waste product. It is called oxygen. Light reaction is divided into two parts. It's basically a phosphorylation reaction, and phosphorylation can be cyclic or non-cyclic photophosphorylation. Light reaction is the phase in which ATP, source of energy, it's a phosphorylated energy rich compound, NADPH2, this is a reducing agent. And oxygen, this is a waste product. Is made. Okay. So the details of cyclic and non-cyclic photophosphorylation I'll discuss with you in the next class, but I'll just give you the headings for that because we need to discuss it in the next class positively. Light reaction or photophosphorylation. Photophosphorylation. Photo means light. Phosphorylation means making some energy rich compound in presence of light. Photophosphorylation is the photophosphorylation, basically you can carry out it as cyclic or non-cyclic phases, is of two types. First is called the Z scheme. Z scheme, or you call it non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Right? The second part is cyclic photophosphorylation. Okay. So in the next class when we meet, we'll discuss cyclic and non-cyclic photophosphorylation and the dark reaction. In the next class, I believe your chapter will be complete. So tomorrow we'll discuss all these things. Okay. So thank you. Have a great day, and please feel free to discuss your doubts with me. I will be very happy to clear your doubts. So I'll see you in the next class, and in the next class we'll finish this chapter. Okay. Thank you. Have a great day, and enjoy your meal.